Hello everybody, my name is Sai Siddiqui, and for my Mitosis video project, I decided to give a little drawing demonstration of what happens during the cell cycle. Yay! And so for the next 5 minutes, you get to listen to my soothing voice and my hands in action as I show you what happens during the cell cycle. And so please mind my little podium assistant, for he will be helping us record this video and any background noise. So sit back, relax, and enjoy the show. So our next part of interphase is, as you were paying attention, synthesis. Synthesis is our second longest phase. Never mind the writing in the back, we'll get into that later. But synthesis consists of 8 hours. And the primary step during synthesis is replication of DNA. And so before we go into replication of DNA, what I forgot to mention in G1 is all the DNA is located in the nucleus in the form of chromatin. Chromatin is basically loose strings of DNA that aren't attached to anything. And so what happens in synthesis is basically our little chromatin is going to replicate. And so this is our little process. So we start out as chromatin, our DNA, and so as a chromatin is being replicated, it then coils around something called a histone. So as it coils and tightly coils around it, it becomes nucleosome. And so then the nucleosome, it coils around and keeps tightening with other nucleosomes, and they eventually form a chromosome. And so all humans contain 46 chromosomes, or 23 homologous pairs. And so I'm sure that is a bit confusing understanding what do you mean chromatin coiling around histones. Well, don't worry, I got you covered. Also, I got a band-aid from cutting myself. So we're going to do a little demonstration with my friend, not tape, sorry, it's a dental floss. Alright, so pretend my finger is a histone, or just a protein wandering around. And so here is our chromatin. Very loose, stringy. So what it's going to do, it's going to coil tightly around my finger. Just like that. And just like that, we have a nucleosome. Except there's millions of these nucleosomes, and they're going to keep doing it with each other to combine and make a chromosome. And so a little bit more into these chromosomes. And so once we have our DNA that is replicated, sorry about the little messiness, is we got now a sister chromatid. Now a sister chromatid is basically 2 identical chromosomes that have the same genes that are attached to each other. And so the attachment point of these 2 chromatids to make a sister chromatid is called the centromere. Basically, and then furthermore, if we follow our little arrow, we have another thing on our sister chromatid called the connectors. Connectors are basically protein complexes, and so in these protein complexes, spindle fibers, which we'll get onto in the G2 phase, be ready for that, they will attach to these connectors and they will pull them apart during anaphase, but we'll get to that later. And so let's get ready for G2 phase. Around for so long, let's get into the final subphase known as the G2 phase. And so the G2 phase is 4 hours long, and so basically what happens is there's some limited organelle synthesis, there's also some protein synthesis, and normal cell division. And so it's pretty similar to the G1 phase, except for one key difference. It'll also just be G1 phase over all over again. It's that the duplication of centrosomes. And so basically, what is a centrosome? So I'll be using the Oxford Dictionary. Kudos to them. And so the Oxford Dictionary defines it as an organelle near the nucleus of a cell that contains a centrioles, specifically in animal cells, and from which the spindle fibers develop in cell division. Thank you, Oxford Dictionary. I will take it from here. And so yeah, that's basically they use everything said. So basically, one of these centrosomes, it will duplicate into another. So another model again. This is a pretty bad model, but this is all I have. Ooh, there's our little chromatin from before. So basically, our centrosome will duplicate to another centrosome. Yeah, so a little McDonald's sign for another one. And so in these centrosomes made of centrioles. And these centrioles are important because these little tubules, they basically make up what is very important, known as spindle fibers. As I said before, these spindle fibers connect to the connectors and are very important for mitosis. Another thing, so into the G1 phase, is once again, we have some more, oh, you can't see that. We have some more checkpoints. Alright, so what are these checkpoints? There's 2 of them now. And so, oh, my mom keeps texting me. If she could stop, there we go. Alright, so these 2 checkpoints are consist of uncyclins. They check to see the DNA is undamaged once again, and if it is fully replicated, as it did, it's in this phase. Luckily, our good animal cell has succeeded in these 2 checkpoints, and I'll go on into drumroll please. Mitosis! Yay! And this is where things really get interesting. So stick around. Right. We are now in mitosis. So mitosis consists of 4 steps. Prophase, our very first. Metaphase, keep on the lookout for a little checkpoint from the cyclins. Anaphase, and telophase. And so there's a pro-metaphase that some people like to include, and we will be including this as part of prophase. And so let's get into our very first one, prophase. Prophase. Alright. So the first thing we need to note is our nucleus dissolves. The most important point, I believe. And so basically, pretend that is our nucleus around there. That'll go away. I can't really get rid of it because it's black. I'm using black. But it goes away. So by the nucleus. And so our centrosomes chromosomes are now visible. And so in our drawing that I'll show you in a few moments, these will be considered our paternal chromosomes, and these will be considered our maternal chromosomes. And so, along with this, our centrosomes will go to the opposite ends of the cell. And so, pretend these are the chromosomes in the middle. Centrosomes are now on the outer ends of this. And so, spindle fibers will continue to form from these. So, these are our spindle fibers, and they're coming from the centrioles. And then, in prometaphase, so basically the spindle fibers will begin to attach to connectors. As I showed you before with the sister chromatid, now lining up. These sister chromatid have these connectors, and they will be attaching to this, which they will eventually be pulled apart. In addition to this, the chromosomes are also beginning to line up in the equator, which is basically the center of the cell. And so, here is my beautiful drawing of prophase. But I'm going to lift up my phone for this. So, prophase. So as you see here, there are maternal and paternal chromosomes. There's only going to be 6, but as you know, there are 46 in all humans, but we're having 6 only for this drawing. And so, pink ones, maternal, these are paternal. And so we have 2 full sets of DNA, because as you know, this is a haploid cell. And so, here are our connectors, and as you can see, our centrosomes, they're at the end of these, with spindle fibers coming out of them, beginning to attach to the connectors. And so, now that this is all done, alright, good. We will now go on into our second part of mitosis, known as metaphase. We are now into our second step of mitosis. And what is that? Well, it is metaphase. Alright, so what happens first is that all the chromosomes, they will line up in the middle of the cell. So you can see here, pretend this is the center. We'll have a more detailed drawing in a second. So this basically is the equator, now in a single file fashion, all the way straight down. And so what this can be described as is a metaphase plate. So here's a cute little pun. This is a plate, and metaphase is written on it. So yeah, it's a metaphase plate. And so all the spindle fibers are also now connected to the connectors, as I said before. So this is all fully done. It was starting in the prometaphase, but now it's completely. And so this is where our checkpoint is. So this is not just a random cube. This is actually our cyclin. And so this will basically check to see if all spindle fibers are connected to the connectors. And so we did do that, so good job, cell. And we will now be able to go on to our next step. But as we can see right now, this is the easiest to identify out of all the steps because of the metaphase plate. So here is a more detailed drawing of what happens on the metaphase. Once again, it will be picked up. This is metaphase. So as you can see, the metaphase plate is being formed with paternal and maternal chromosomes being lined up single-filedly. Our centromeres, the centromeres are at the ends. All the spindle fibers are developed and they're connected to all the connectors. And as our cyclin has completed our checkpoint. So now that we are done with that, we are ready. There we go. We are ready for our next step, which is anaphase. We are now in anaphase. And so anaphase basically starts out with the sister chromatid being pulled apart. We're going to have a little live demonstration. So pretend this pencil, they form a sister chromatid. Alright? So the exact same genes on each side. Alright? It's not meant to be different colors, but yeah. So basically, our duct tape, our spindle fiber, will attach to the connector, alright? Right there in front of it, and it will pull one part of the sister chromatid away, and we'll pretend this is another sister chromatid, we'll pull this part away. And so now the pulled apart sister chromatid is now referred to as a daughter chromosome, where it will contain the exact same genes as the other one. And now both sides, and two new cells that will be formed, known as daughter cells, will have two exact same copies of chromosome. And so let's go back into anaphase. And so along with the sister chromatid being pulled apart, the connectors begin to disassemble the spindle fibers piece by piece. As I said, little protein complexes, they'll get rid of the little stringy things or spindle fibers attached to them. And then finally, the polar spindle fibers will extend outward. Now, keep in mind, polar spindle fibers and spindle fibers, these are not the same. It's a related way, but basically, as I told you before, spindle fibers, they will connect to the connectors and pull apart the sister chromatid. This is very different from the polar spindle fibers, where they don't do this at all. Instead, they push the membrane outward, allowing the cell to widen, and so that two new cells will be able to form. And so once again, we have another drawing of anaphase. So here it is. So here we see an anaphase. The cell, as you've seen before, is no longer a circle or oval as before, for it is now being pulled apart due to this polar spindle fibers. They're now pushing each side outward. And along with this, the sister chromatid, now daughter chromosomes, are now being pulled apart. As you can see, one strand, two exact same. They are the same genes, same alleles, but they're now separated and now referred to as daughter chromosomes. And this happens with all the chromosomes that are lined up at the metaphase plate. As they're going on, along with this, the connectors are now beginning to disassemble some of the spindle fibers. And so now that we are done with anaphase, we are on to our final phase, telophase. Alright everyone, so unfortunately we are at the end of the video. It's been a lot of fun. Just want to thank you all for being here for so long. I know cell cycle may not be the most interesting, but hopefully I made it a little fun for you guys with this little drawing. So let's get into our final phase of mitosis, and we'll end off with cytokinesis. So our final phase, if you've been paying attention, is telophase. Telophase is pretty easy to understand. Basically, our spindle fibers, things that are connecting to our connectors and pulled apart the sister chromatid, have now fully dissolved. And in addition with this, new nuclei form. So in our single cell still, all the new daughter chromosomes that have been pulled apart, there are now nuclei formed around them. So once this happens, cytokinesis is ready to happen. So cytokinesis can happen in two different ways based upon the cell it's happening. So we'll begin with the animal cell. So what happens in the animal cell is basically a contractile ring forms. And this contractile ring is basically made of actin filaments. And so these basically pull in the cell and the membrane, and this pinch of the cell is known as a cleavage furrow. And so this can't happen though with a cell plate because it is a square. And it's not a square, is because of the cell membrane of the shape. It's a square because of the cell wall causing its rigidity. And so because of the cell wall, we can't exactly have a ring pushing in, so instead we have something called a cell plate. So here's a cute little pun again. Cell plate. We erased our metaphase to make a cell plate. Alright, so in our cell plate, basically, a new cell wall forms in the middle, and it's inward and pushes outward, and basically this will separate, making two new daughter cells. So let's show basically what happens. We're going to start with we're going to show an animal cell. So I'm going to lift it up for the last time. Telophase and cytokinesis. So as you can see, our new nuclear form, we have no spindle fibers left. Those are all gone. Same thing with other cell. And so now there's a pinch in, all because of the contractile ring formed from the actin filaments, known as a cleavage trail. And so once this continues to push in with the pinch in, eventually they finally separate, making two new daughter cells. And then these two new daughter cells will go through the same cycle. So as I showed you in the very beginning, they'll go from their G1 phase, then to their synthesis, then their G2, and then finally mitosis again, furthering this. And so as you can see, the whole point of the cell cycle is just further grow cells and allow it to regrow tissue. And so as you can see, we are at the end of our cell cycle. And so I just want to thank you all for being here, and we'll end out with a little outro.